this is this structure here. This is the urinary. So, actually, not here, but this structure here is a urinary bladder. We're looking at it from its posterior surface, and these two white structures that you see here represent the are the vas deferens. The vas deferens are these tubes that you see coming up here. There's the scrotum, the testes. There's a tube that comes up and it goes around the back side, each side of the bladder, and that's what these are. The vas deferens deliver sperm to the ejaculatory duct. So sperm cells, they're being pushed through muscle contraction. It's the only way they can get there right now, because sperm cells have to swim in a fluid. Now the fluid is going to come in from these two sort of pink structures here. Those are the seminal vesicles. Now the seminal vesicle and the vas deferens combine to give you what's known as the ejaculatory duct. You can sort of see it on the illustration here. There's the ejaculatory duct. There's a seminal vesicle on each side and a vas deferens on each side, and they come together so that now the sperm have a medium to swim in. Is the semen. 60% of the semen is generated by the seminal vesicles. Another 30 to 35% is going to be created by the prostate gland. And then the last 5% comes from this gland called the bulbourethral gland. The prostate gland is directly below the bladder. So this little round structure here you see on the illustration. Here's the urinary bladder up here. There's the prostate gland there. There's a seminal vesicle. There is one of the vas deferens. And this is the ejaculatory duct. The two ducts come together, or maybe just one big duct comes together and discharges into the what we call the prostatic urethra. That's the urethra drains the bladder, but the prostate is right below the bladder here. It's about the size of a walnut when it's healthy. And the urinary bladder, urine will go through here. During ejaculation, the prostate has a sphincter right there at the tip of the urethra, the closed valve, so no urine comes out during an ejaculation. So the prostate gland is designed to produce semen also. About 30% of the semen is generated there. The last 5% of the semen is generated by what we call the bulbourethral gland. It's this little tiny structure. You follow the urethra back. There's a gland. This is the bulbourethral gland. This is the first 5% that's generated. The bulbourethral gland will secrete semen prior to ejaculation. It will cleanse the lining of the urethra, and it will also lubricate the glands, the tip of the penis. So that's what its role is. Older books call it the Cowper's gland, which is probably the name of the anatomist that discovered it. But a more precise term would be bulbourethral. So harder to remember too. Now, the delivery mechanism is through this urethra that's found inside the penis. Now, the penis contains 3 sets of what we call spongy tissue that are designed to become engorged with blood and fill up. There are 2 bands, 2 circular columns of this spongy tissue on the dorsal surface, the top surface of the penis. And they're called the corpus cavernosum. Surrounding the urethra is another layer of spongy tissue called the corpus spongiosum. Now, the spongiosum also becomes engorged with blood. It fills up, and between the three tubes, it causes the penis to become erect when the male is sexually aroused. The spongiosum is there to protect the urethra so that it doesn't get bent or twisted or damaged in any way. So the testes are suspended in a sac below the abdominal cavity, outside the abdominal cavity. They're connected, to the body, but they're suspended in a sac. And the reason for that is that the testes this is the site of sperm production here. Sperm mature are developed in the seminiferous tubules. You saw those last week when we were looking at where testosterone gets made. And sperm cells that are manufactured, that are created there, have to go into the structure called the epididymis for about 3 weeks to mature. The testes are designed to keep the sperm cells at a slightly cooler body temperature. It seems that the enzymes on the head of the sperm can't survive at body temperature 98.6. And so what'll happen is the enzymes will become non-functional if they get too warm. They're very sensitive proteins. So by suspending the testes below the abdominal wall in a separate sac in the scrotum, the temperature stays around 94 degrees, and the enzymes at the tip of the sperm aren't destroyed. Now, why do they do that? Well, it seems that the egg in the fallopian tube has 2 distinct layers that surround it. And the only way to get through those layers is to dissolve them using the enzymes contained inside the tip of the sperm. If the enzymes don't function, you can have all 400 million sperm during an ejaculation up there at around the egg, and nobody's going to be able to get in. You have to be able to digest away these 2 protective layers around the egg. So, and I said 400 million sperm. Typical ejaculation releases about 400 million sperm in semen. By the time the sperm cells reach the egg, if there's an egg there, reach the area where the egg should be, they're down to about 2,000 or 3,000 sperm cells. There's a 99.99% mortality rate inside the female reproductive system. So, okay, this is a cadaver cross-section in here. Someone asked yesterday a really good question: how did they get such a precise cut? And either the cadaver was frozen and they used a very, very sharp multi-bladed power saw to cut the body in half, or they used a laser. But this is not something you could dissect out that precise, at least with a scalpel. Now, a couple things you can see here. You can see the spongy tissue in the penis. The cavernosum is on the dorsal surface. That's the top surface. The spongiosum surrounds the urethra. You can see the testes and the epididymis here where the sperm goes to mature. You can see the urinary bladder right here. And this is the prostate gland right here. And it doesn't look like much. A lot of these cadaver images, it all looks the same, in tissues anyway. However, there is one thing that's pretty important here. You'll notice just how close the prostate gland right here is to the rectum. And this becomes a very important diagnostic tool, this location. There's an examination called a digital rectal exam. It sounds like it involves technology because we use the magic word digital, right? In this case, digital means finger. And so when a male has a prostate exam, the physician or whoever is doing the examination will insert their finger up the rectum and push up into this area of the rectum up here and palpate the prostate, feeling for a lump. And it's a very effective technique, but it doesn't tell you if there's cancer or not. It just tells you there's a lump, or it tells you that the prostate's enlarged. The definitive test, of course, is to have a biopsy. So when a male has a prostate examination, yes, there is blood work that can show you, the PSA test, but the finger exam is much more definitive for this. Your average family practice physician may not be able to detect the more subtle growth taking place in a prostate, simply because, how many digital exams does a family practice physician get to do on a weekly basis? A handful, if that. Whereas your typical urologist would be doing a lot, 10, 20 every day. That's what they're trained for, is to be able to discern what, that there's a lump there or not. So whereas your family practice physician could say, well, I don't know, there might be something there. The urologist will say definitely yes or no, and then that will lead to a biopsy. So we have a nice slide in lecture actually showing from this perspective of a cadaver, showing you exactly, someone has placed their finger in the rectum of the cadaver, showing exactly where they would be palpating for. So it's not any different than self-examination for a lump in the breast. So, okay. Now, if we take a closer look here, we got a cross section of the penis. We can see the top two spongy tissues, the cavernosums paired there. They run the length of the penis. What happens is when the male becomes sexually aroused, blood flow will increase, and the vessels leading into into the penis will dilate. That means they'll get bigger. They'll vasodilate. Blood flow will increase in those tissues, and the more blood that goes in there, the more it will compress the veins that are normally used to drain these areas. This, our circulatory system is designed so arteries bring blood in and veins take blood out. Well, what happens here is blood flow increases going into the spongy tissue when the male is sexually aroused. Spaces fill up with blood, they compress the veins and restrict the flow coming back out. And so the process of sexual arousal, and I don't want to get into the physiology of it, but it stimulates the release of a chemical known as nitrous oxide. Nitrous oxide is a very powerful vasodilator, meaning it causes our blood vessels to dilate. That causes the vessels there in the spongy corpus spongiosum to dilate. More blood comes in, presses the veins trying to come back out, and so an erection occurs in all three areas there. Now the testes are sitting inside the scrotum. This is the scrotum here. Inside here we have a series of muscles and blood vessels and nerves. Here of course we also got the vas deferens. So the two sets of muscles that are pretty critical here are the dartos muscle, that's this layer right here, and the cremaster muscle. The cremaster muscle is a very long muscle. The dartos muscle causes testes to wrinkle or to relax. The cremaster muscle will draw the testes up near the body on a cold day or in cold temperatures. It will allow the testes to relax and move away from the abdominal wall on a hot day. So the cremaster muscle is actually a temperature regulator along with the dartos. Now, you see the epididymis surrounding the testes itself. You can see it on the right-hand side. There is a very specific network of veins and arteries here. One of the problems in males is that males make sperm cells. Males make sperm cells at the start of puberty, and they make them till the day they die, 80, 90 years later. Now, those sperm cells have never been identified by the T cells. Our T lymphocytes, the ones that go into the white cells that go into the thymus gland to become immunocompetent. So they're trained to identify foreign antigens. Well, their training takes place in the first year of life. So by the time males are 1 year old, all their white blood cells, all their T lymphocytes have been trained to recognize foreign antigens and kill everything and not attack self, not attack our own antigens, except that sperm cells weren't around at 1 year of life. Sperm cells don't start forming until puberty. So the sperm cells, as far as the T lymphocytes would be concerned, are foreign substances and must be destroyed. So we don't let the T cells get into the intestines. We keep the T cells away from the testes so that we don't lose sperm cells by being attacked, having an autoimmune response. You're attacking the sperm cells. The T cells never get into the scrotum and into the testes because they see sperm cells as foreign. Now, females don't have to worry about that. The egg cells that are produced every month from the ovary, they've already been there. They've been there since birth. They've been there since before birth. So the woman's T lymphocytes and B lymphocytes recognize the antigens as self, and so there's no problem there. During intercourse, when sperm cells are released into the vagina and the uterus, they're fair game for the immune system of the female. They can be attacked by T cells and B cells because the sperm cells are foreign antigens, and they do get attacked. As I said, the mortality rate is extremely high. Okay, now there's a scrotum holding a testes. They're paired, and the scrotum contains both of them, but they're in two separate pouches, if you will. Okay, this is a cross section of the penis. Down here at the bottom of the slide, you can see we have the spongiosum surrounding the urethra. That is the urethra. There's the corpus spongiosum. There's the urethra right there. This, there are three parts to the urethra. We have the penile urethra, we have the membranous urethra, and we have the prostatic urethra. But here we are protecting the urethra with this layer of spongy tissue that will firm up when it becomes engorged with blood. Here is the corpus cavernosum up here. There are two layers of that, two tubes, one on each side, engorged with blood, causing the erection to occur. And here we see what it looks like there. Now, this is a better picture. So, and you can see the urethra right above the words spongiosum. That little opening on the side there is the urethra. So we have these three layers here within the penis. Now, they will only become engorged if blood flow, if the vessels supplying the spongy tissue become dilated. So, sexual arousal in the male releases nitric oxide in the arteries and arterioles in the cavernosum. That's what increases the blood flow. We'll talk a lot more about the physiology of that in lecture. Okay. Here we see the cross section of the testes. You saw the testes last week, but you can see here, this is where these are the seminiferous tubules. This is where sperm production takes place inside the testes. These are all immature sperm cells down here. In the lumen of the seminiferous tubule, and there are hundreds of these tubules. Sperm cells form from stem cells known as spermatogonia. Spermatogonia, whenever you have the term gonia at the end of a word, it usually means that it's a stem cell, and these stem cells will produce any cell we want. In this case, these are going to be producing sperm cells. Now, these spermatogonia along the inside lining are diploid cells. That means they have, all 46 chromosomes. Sperm cells have to be haploid. They have to have 23 because it has to combine with an egg to give us 46 ultimately in the baby. What will happen is, like a lot of sperm cells, a lot of stem cells, spermatogonia will undergo mitosis. Half of them will stay right there along the inside lining as spermatogonia. We call them, type A spermatogonia. Those stay there because they get to do it, reproduce again and again and again. The other half of the daughter cells are going to be type B spermatogonias, and they will start undergoing meiosis. Now, meiosis is nuclear reduction. With meiosis, we go from 46 chromosomes down to 23. And every type B spermatogonia will form 4 sperm cells. And so those sperm cells migrate towards the lumen of the seminiferous tubule and then ultimately move. They are immature sperm cells there in the lumen. They're called spermatocytes, and then ultimately they are spermatozoans. Once they are mature enough to move, they will move into the epididymis, and for another 21 days they will be developing and growing, waiting to be ejaculated. If there is no ejaculation after they're in there, then they will start breaking down and be reabsorbed. Another, again, this is a seminiferous tubule. There in the lumen, we have lots of spermatozoans. There's your spermatogonia along the inside lining. Our spermatocytes growing as they move, as the spermatocytes migrate towards the middle of the tubule. They are busy undergoing meiosis. There's 2 stages to meiosis, and they ultimately end up with 4 spermatozoans inside the lumen. Each one of them are going to be haploid. They're going to have 23 chromosomes. But the most important chromosome they carry, though, is going to be an X or a Y. Sperm cells each carry a sex chromosome. Now, in the males have an X chromosome and a Y. This is the female chromosome. This is the male chromosome. Sperm cells are either going to be X or Y. And so it is the sperm cell, it is the male that determines the sex of the baby. Because the egg, this is an oocyte. That's an egg. Okay. There, it's an egg. Now it's an egg. Are always X, because the only sex chromosomes in the female's body are X. So egg cells are always X. Sperm cells can be X or Y. If a Y sperm fertilizes an X egg, it's going to be a male. If an X sperm fertilizes an X egg, it will be female. Now, granted, the genetic information provided in the nucleus of the sperm cell are also going to be, give you dominant and recessive traits, just like the nucleus of the egg. So, hair color, eye color, skin color, important factoids like dangling earlobes and can you curl your tongue, for whatever reason, but also things like, are you going to have blonde hair and blue eyes? You're going to have green eyes, you're going to have brown eyes, you're going to have brown hair, you're going to have red hair. How come you have your grandfather's nose? Because it looks just like your grandfather, or you have curly hair, just like your all your aunts did. So it's because of the genes that are being passed on here. But the one thing that that the sperm cell does decide is going to be the sex of the baby. Now, this is a trick question, because I'm tired of people getting it wrong. When is the sex of the baby determined? It's not that difficult. Just think. Fertilization. Yes. Bingo. You win the prize. You get to stay for the whole class.
I was gonna do that anyways.
I know, so there you go. So, okay, yeah, the moment that the sperm cell, the nucleus of the sperm cell fuses with the nucleus of the egg cell, the sex of the baby is determined at that point. So right at fertilization. Yeah, lots of people over the years have always marked down 3 weeks after conception. Close, but no, it's right at the point of conception, because the sperm, the sex chromosome is there. And as soon as that nucleus of the sperm cell fuses with the nucleus of the egg cell, we have our sex determined. So now we won't know that for weeks and weeks and weeks. We have a grand something on the way from and we don't know yet if it's a grandson or a granddaughter. And it was 9 weeks at Christmas. So and my son said they still haven't been able to, until she's a little bit older, they can't do the testing to determine if it's male or female, but they'll know soon. So at Christmas, it was a 9-week-old something. It was a grand something. So anyway, stay tuned as I hear more on that. I've got a great picture of him or her at 9 weeks. So, but the point is though, that we won't know for a while, but the baby knows. The baby will start developing almost immediately all the characteristics, all the anatomy of reforming. So we'll know soon. Anyway, but at the point of conception is when the sex is determined. Okay, now, okay. Sperm cells. Hundreds of millions of sperm cells get released during ejaculation. Only a few thousand make it up to the fallopian tube where the egg is located, if there's an egg there. Sperm cells contain the head structure contains the nucleus. So the nucleus is going to be found right up in here. Nucleus is in there. There is a structure here called this is the head. This is the mid piece right here. This is the flagella or tail here. And there's a cap on the sperm called the acrosome. And that's where the digestive enzymes are located. Okay. Now the mid piece is right behind the head. There's the tail or flagella. There's the acrosome. You can see it a little better on the slide there than I drew it. And there is the mid piece. The mid piece contains lots of mitochondria for energy. Because if the sperm cells, as soon as ejaculation occurs, they're going to swim as fast as they can. And they're going to use that flagella, that tail, to whip back and forth to move through the semen.
And it's similar to like a tadpole, sort of.
Yeah, it looks a lot like the swimming motion, sort of like a tadpole, except that sperm cells have no reverse gear. They go forward and they get stuck. They get stuck in all the infoldings of the female reproductive tract. The vagina, it's always like, well, the vagina is invaginated. Which came first? Which word came first? Meaning it has lots of folds, and the sperm will swim into these folds and get stuck. They'll keep bouncing against the folds, and maybe they'll bounce loose and move around, but it's very difficult for them to once they get stuck, to get unstuck. And so they use up all their energy trying to keep swimming forward. The ones that do get into the uterus, a fair number of them, they've got two choices. They can go to the left or to the right. And a fair number of them may go the wrong direction. There may not be an egg on one side or the other. And even then, if there is an egg, it may have been too early. Maybe the egg hasn't been released yet. Maybe the sperm's there and the egg hasn't ovulated from the ovary yet. Or maybe the egg has already gone past its window of opportunity. So lots of things can happen. But Sperm cells will spend a great deal of effort and energy swimming as hard as they can to get wherever they're supposed to, they're trying to go. The acrosome does a very important job. The acrosome protects males and females from those digestive enzymes. Keeping the digestive enzymes contained in the acrosome is so they don't eat away tissues. They don't dissolve tissues in the testes, in the scrotum, in the vagina, in the uterus. They're designed to break down the layers of tissues that surround the egg. We don't want, so it's a very specific time when they can get released. The acrosome has to actually sort of break apart before it can start releasing those enzymes. Okay. Now this is the prostate gland. The others, 30 to 35% of the semen that is produced comes from the prostate gland. You can see here in this model, the prostate gland is this structure, this little structure down here at the bottom. I'll pass this around. And it's about life size. You can look at the pictures here. This is the urethra. This big structure here is the urethra. And then these are the glandular follicles up there on top that produce the semen. This is a perfectly normal looking, healthy prostate. The follicles are all more or less reasonably shaped. There's nothing irregular. There's not a lot of extra cells around there that shouldn't be there. The follicles themselves look reasonable. A lot of them are about the same size. There's not a lot of irregularity. On the other hand, prostate cancer, there's the urethra, smooth muscle around the urethra, and here are all the follicles that are going to be filled with semen produced by these glandular cells along the sides here. Okay, now the prostate has several things it can affect. Normal prostates look like that, look what we just saw. On the right-hand side, upper right, we have something known as benign prostate hyperplasia, BPH. It's an enlarged prostate. Lots of older males develop BPH. The prostate swells and grows. Don't know why. It's not cancer, but what it does is it grows and starts narrowing the urethra. So a man with BPH can't empty his bladder when he pees. And so they say he doesn't have a good stream, a good flow. And so there's always a constant need to urinate. So we have this highly enlarged prostate gland. There's no connection between prostate cancer and benign prostate hyperplasia. Hyperplasia simply means excessive cell growth. The cells get bigger. It isn't so much that the cells are reproducing rapidly, but they're just enlarging. That's what hyperplasia means. And so you treat it with drugs, you treat it with surgery to keep the urethra open, because it could be like, every hour you'd have to, if you have an enlarged prostate, every hour you'd have to wake up to go pee during the night, and that's very difficult. On the bottom left is prostate cancer. I got some more slides to show you on that, but prostate cancer is, like any cancer, is really ugly looking. You see a lot of overgrowth. Cancer cells don't have contact inhibition. They will continue to grow on top of each other. Instead of having very large cells, you'll have lots of little cells all in sort of an undeveloped state. They'll grow and they'll reproduce, but they won't ever really grow into the shape they're supposed to, and you're going to have many, many cells there. And so prostate cancer will start out in one region of the prostate, and it will continue to grow and spread to other regions. And then from there, it can metastasize. It can spread to the bone and muscles around the prostate. And from there, it can spread to any other part of the body. And the third category here is, and the last one, is prostate inflammation or prostatitis. Just like any other organ in our bodies, the prostate can become inflamed. It's been irritated by something, and it is inflamed, it has engorged blood flow there, it's swollen, it's tender, it hurts to pee, things like this. Individuals that maybe don't urinate as often as they should, that are trying to hold it, because after all, the urinary bladder can rupture, believe it or not, if it is filled with too much urine and the pressure isn't relieved. It takes a lot of pressure to rupture that structure. But inflammation of the prostate, any number of things could cause that. So usually very treatable. Maybe it's a bacterial infection. Maybe you have to treat it with anti-inflammatories, but if it's probably pretty painful too. Okay. And this is what the enlarged prostate does. It squeezes and compresses the urethra. That's what makes it difficult to urinate in here. And here we see the difference between normal healthy cells on the left and cancer cells on the right. Notice how they're disorganized. Notice how they just sort of grow all over the place on top of each other. Healthy cells practice contact inhibition. They don't grow on top of each other. When there's a cell there, a new cell won't grow right beside it. But here, they're all over the place. The follicles are going to be irregular in shape. They're not going to be doing their job. And if you could look carefully in this, you'd probably see lots of cells in various stages of mitosis also. But here, they all look about the same because they're all in about the same stage of the cell cycle. Okay. In the female system is a little more complicated. We take a look first at the uterus. The uterus is the structure that receives the fertilized egg or not. It's about the size and shape of a pear, an inverted pear. And it is held in place by several large ligaments, the ovarian ligament and the round ligament, and various other muscles are attached here to hold the uterus in place. The uterus is tilted at a 90-degree angle directly over the urinary bladder. In the female, it's tilted forward over the urinary bladder. There are two fallopian tubes, one extending out on either side, fallopian tube, oviduct, and uterine tube, if you learn it as fallopian tube, call it fallopian tube. Only the newer books call it the oviduct or the uterine tube. Doesn't matter. It's got three names, whatever works. The fallopian tubes extend out about 2.5 inches away from the uterus, and then suspended below the fallopian tube is the ovary itself. The ovary, each ovary is attached to the body of the uterus, but there's no direct connection. The ovaries are about the size and shape of a grape. So you have these little structures on either side, and each month, every 20, the ovarian cycle runs through a 28-day cycle, and on the 14th day of the ovarian cycle is when ovulation occurs. So doesn't matter what day it is in the ovarian cycle. If your period started on January the 1st, by January 14th, if that was the day that your ovary ovulated an egg, that's ovulation day. If it was the 17th, that still becomes day 14. Doesn't matter what day ovulation takes place, the events from ovulation to the new menstrual cycle is going to be 14 days. So now a couple structures here are pretty important. These things here are called fimbriae. Fimbriae means fingers. This is a model of a very large ovary right here. And this is the end of the fallopian tube. And these fimbriae extend out here, and the opening of the fallopian tube is inside the fimbriae. So what happens is that the egg gets released at ovulation. The egg gets released into the abdominal cavity. Now it can go anywhere. It doesn't have to go into the fallopian tube. But these fimbriae, they look sort of like anemones. All we need now is a clownfish in there. But the fimbriae create a current to draw the egg in and draw it up into the fallopian tube. Now, in lecture, we'll talk about all the things that can go wrong with that. But that's what's supposed to happen is the fimbriae create a current, the egg is drawn into the fallopian tube, and then smooth muscle contractions and cilia lining the fallopian tube will push the egg along down towards the uterus. Now, fertilization, if fertilization takes place, occurs up here in the ampulla. In this first bend up here is usually where sperm cells and the egg are going to meet. If there's not enough sperm cells, likely fertilization won't occur. If the sperm cells got there early, the egg hasn't been released yet, those sperm cells will continue on out into the abdominal cavity. If the egg's been released and it's further down the tube, the egg is only viable for about 48 hours, and the sperm cells get there a little later, then conception probably won't occur either. There's really only about less than a 20% chance that successful fertilization will occur in here. Now, regardless of that, the egg will continue to move down the fallopian tube to reach the uterus. Now, that's a 2.5-inch long trip. It takes 7 days. And if the egg has been fertilized, it may or may not decide to implant. It has to implant in the inner wall of the uterus. No guarantees it's going to implant. If it does implant, it implants around the 21st day of the uterine cycle. Now, the uterine cycle only lasts 28 days. At the end of the 28th day, menstrual flow is going to start, and that's going to be day 1. So if that egg implants, that fertilized egg implants in the uterus on day 21, it needs to move quickly to start telling the ovaries and the uterus that, hey, I'm here, I'm implanted. Don't cause menstrual flow to occur in 7 days. So if implantation is successful, and again, most times it isn't. Okay, so here is another one of those cadaver cross sections. Here you can see at the top up there, we see the uterus. The uterus is tilted forward. Here's the uterus tilted forward. There's the urinary bladder right there. So you can imagine the pressure on the bladder during pregnancy when you got the baby growing inside the uterus, and you got the amniotic sac, and you got about 25 pounds of baby and fluid pressing against this uterus constantly, because the other side of the uterus is, of the bladder is the pubic symphysis. So, talk about between a rock and a hard place. You got the pubic symphysis on one side, and 25 pounds of baby and fluid on the other side pushing against that bladder. Your female, as she gets in her later stages of her pregnancy, going to have to pee a lot because of this. So you also see, you can see this long tube here is the vagina leading up into the uterus. You see the uterus is tilted again 90 degrees over, and then the urethra right in front of this here from the urinary bladder, and you can barely see 2 folds of tissues here. One's called the labia minor and one's called the labia major. Drawing will show it a little better here for you. Okay. So here we see typical uterus. tilted at a 90-degree angle over top of the urinary bladder. You can see the vagina coming in from underneath. This is the front part of the body here. This is the dorsal surface. This is the ventral surface, or anterior or posterior. This is structure here is cervix. The cervix is the opening to the uterus. And then we have the vagina here. Here we have the labia major, the outer fold. The labia minor surrounds an area called the vestibule. The vestibule contains both the vaginal orifice and the urethra, the urethral orifice right here. And then at the at the anterior end of the vestibule, you have the clitoris right here. Now the clitoris is made up of spongy tissue, just like the cavernosum and spongiosum in the penis. When the female is sexually aroused, the clitoris will become engorged with blood and become erect also, not to the extent that the penis will. But the clitoris, the tip of the clitoris and the glans penis, the head of the penis, share one unique trait. They are both extremely sensitive to touch. The glans penis, the tip of the penis, and the clitoris are both 10,000 times more sensitive to touch than any other structure in the body. And so it is this mechanical stimulation of the clitoris and mechanical stimulation of the glands, of the tip of the penis, that causes ultimately orgasm in the male and female structures here. So now directly behind the vestibule, we have the anus in here. We see the anus is right here. Anal opening, vaginal opening, urethral opening. This location of all three of these openings contributes to a problem that many women have with urinary tract infections, because you have this area that is usually warm, dark, and moist. Bacteria can migrate from the anus, regardless of how clean someone is. Bacteria can become present and move into the urethra and cause a urinary tract infection. You'll notice that the distance, the length of the urethra here is only about 5 centimeters. There's the urethra right there. So it's very easy for bacteria to migrate up into the bladder and cause an infection. Males tend not to have urinary tract infections very as frequently. If it can happen, it occurs much less frequently in females, simply because the urethral tract in the male is about 20 centimeters long. So it has to run all the way up, through the penis, up through the prostate, into the bladder. The distance gives the male's immune system time to fight an infection. So while males may be exposed to the same type of bacteria, they're less likely to become symptomatic because their immune system has had time to fight it off. Whereas in the female, the immune system is, by the time it's activated, the bladder is already infected and you have a UTI going on there. And simply because of the anatomy of the female reproductive structures that make this a more frequent problem, if you will. You'll notice the ovary suspended on either side of the uterus, and the uterine tube will then discharge into the uterus. If this were if the top of the pear, there's our uterus right here. The fallopian tubes would come in sort of at this area right in here. And just to make things a little more challenging, the tube narrows down a lot as it moves into the, let's make it smaller. There we go. It narrows down as it gets into the uterus too, just to make things more challenging for the egg. So, okay. The ovary contains all the eggs let's just rephrase that. Conventional wisdom says the ovary contains all the eggs that the woman is ever going to have. Typically, somewhere in the neighborhood of several million eggs are are laid down while the female baby is in utero. Somewhere around the neighborhood of about a million are available at birth. At the onset of puberty, there's something in the neighborhood of 400,000 eggs available to be released. Now, these eggs, many of these eggs are kept in a very primitive state. We have the immature eggs tend to be found along the inside edge of the ovary, and every month a handful of those eggs will come to maturity, and one will usually be released. That's conventional wisdom. We'll talk about this in the lecture, but there is some speculation that not all the eggs released by the ovary are as old as the woman is. That eggs may be being generated from the ovary. So it just we'll talk about the questions behind that. Anyway, a couple terms to sort of try not to get hung up over. We have what's known as a follicle and an oocyte. A follicle is the container that holds the egg. The egg is called the oocyte. So here we have a primordial follicle. This is a very, very immature follicle containing an immature oocyte. Now, over the course of the reproductive lifetime of the female, the ovaries are going to be saturated with follicle stimulating hormone. It stimulates the follicle to grow. The oocyte, the egg inside the follicle, is exposed to estrogen, and it continues to grow. And every month, one egg usually gets released through ovulation. Now, what happens is that the egg that gets released, let's say today is the 14th day of the cycle, the egg that gets released was actually pre-selected a year ago. It and a handful of its closest friends were brought to maturity. They were saturated with constant doses of follicle stimulating hormone, and they were all brought along. They all matured, and one of those eggs was selected as to be the one that was ovulated. The rest of them disintegrate. And so this egg was getting ready to ovulate here, and when it's popping out here, they were pre-selected a year out. And so every 28 days, an egg will ovulate on the 14th day of the cycle. There's no rhyme or reason as to which ovary it comes from. They don't alternate. They may alternate, don't know. They can all come from one ovary. They can come from both ovaries. They can take turns. It doesn't matter. But generally, you'll have one every 28 days. You probably about 13 eggs ovulated a year. And this will continue for 40 years, 30 years, somewhere in that neighborhood. And of those 400,000 eggs, you'll generally see a half a dozen eggs every month being involved in the process to select the egg of the month, if you will, or the egg of the cycle. And the conventional wisdom says that if a 40-year-old female gets pregnant, she got pregnant with a 40-year-old egg. And an old egg like that has problems with, it's old. The DNA may have gotten fragile and damaged over time. And, there's always a concern of some sort of birth defect in the baby because it's a 40-year-old egg. But we don't have a lot of, the numbers don't tend to match up with that. We don't have more cases of birth defects from 40-year-old eggs like we should. So there is a lot of thought going into this, a lot of studies that says that maybe the female ovary is generating newer eggs, younger eggs during her reproductive lifetime. We'll talk more about that in the lecture. So anyway, day 14, the ovulation occurs, the egg is released. It then starts drifting up into the fallopian tube, and then the old graphene follicle. That's this structure here. This is called the graphene follicle. This is a mature egg getting ready to be ruptured and released through ovulation. The old graphene follicle becomes the corpus luteum. The corpus luteum is busy secreting progesterone. Progesterone keeps the lining of the uterus intact. And it will continue to secrete progesterone up until about the 26th day of the cycle. And then it'll shut down. And that will cause the uterine lining to shut down and start sliding out on day 1, about 2 days later. Unless, of course, the egg has implanted itself successfully. The fertilized egg has implanted itself, in which case the fertilized egg will start producing a hormone called HCG, human chorionic gonadotropin, which signals the corpus luteum to keep making progesterone. HCG is what shows up in pregnancy test kits. The early pregnancy test kits, they detect the HCG in urine, and if HCG is being released, then it tells the corpus luteum to keep producing progesterone, which keeps the lining intact. No implantation, no HCG. Corpus luteum shuts down after day 26, and menstrual flow will start in 2 days. And this can occur even if implantation was successful. No guarantees on this. Okay, there's what your graphene follicle looks like. You have your egg right here in the center. This is called a secondary oocyte. Right there. And it is surrounded by, there's the nucleus. There are 2 layers of tissue. One's called the corona radiata, and the other is called the zona pellucida. Corona radiata is the outer layer, zona pellucida is the inner layer. These are the 2 layers of tissue that have to be dissolved away by the enzymes in the acrosome. And so this graphene follicle is very close to being released. It has a mature egg. It has its protective covering there. It's ready to go. And now, these are the immature cells. In here, there's the lining of the ovary, and all those little round things there are follicles with immature eggs inside there. And those eggs, there they are. These immature follicles and the immature eggs will continue to develop because they're going to get saturated with follicle stimulating hormone every month released excuse me, by the anterior pituitary. And they're going to continue to grow and grow, and some are going to get selected to be released a year later as they mature. And this process will occur on a 28-day cycle. The ovarian cycle and the uterine cycle run together. Each is a 28-day cycle. And, no guarantees. No guarantees that fertilization is going to occur. No guarantees that implantation is going to occur. No guarantees. Okay. There's your corpus luteum. It secretes the progesterone that will hold the lining intact if it's being told to by HCG. Now, the corpus luteum will release progesterone, and it will do it up until about day 26. That gives the fertilized egg time to start the production of HCG. If HCG production isn't occurring, then it'll shut down. That's the check and balance on this. If implantation was successful, the corpus luteum will continue to produce progesterone for about 3 months. After 3 months, the placenta takes over the job and the corpus luteum shuts down. The placenta will continue to release progesterone right up until time for delivery, because that's what's holding the uterine lining intact. Okay. And you saw this last week. This is the corpus luteum. That is the large one there, that large round structure on the top of the ovary is the one that's been secreting progesterone. Those are scar tissues. The other brown areas and white areas are scar tissues from previous corpus luteums. Yeah, because once ovulation occurs, then the remnants of the Graafian follicle will start doing this, and it will do, it will produce progesterone at least till day 26 of the cycle. It will shut down then if there is no HCG, but if HCG is present, it will continue doing its job for 3 months. Okay. There's your uterus right there. The uterine lining we have the innermost layer is called the endometrium. That's the layer that the fertilized egg will implant in. The outer line, there's your lumen. The outer lining of the uterus is called the parametrium. Underneath that are 2 layers of smooth muscle called the myometrium. And then underneath that is the endometrium. The endometrium has 2 distinct layers. It has a stratum basalis, which is the basement layer. The stratum basalis puts right up against the myometrium. And then the stratum functionalis grows off of the basement layer. The stratum functionalis is a layer that will build up and become engorged with blood, ready to receive a fertilized egg and let it implant, maybe. So that's also the layer that slides out during the menstrual flow. Okay. Here is the lining of the vagina. The outer layer of the vagina is covered with stratified squamous epithelial cells, the same type of cells that we found on the inside of our mouth. And these cells are going to be generating lots and lots of mucus, but there's multiple layers there so that wear and tear can take them off. That's what stratified squamous cells do. And underneath all of this is layers of smooth muscle. The stratified squamous cells are different from our skin. Our skin is stratified squamous, but our skin is waterproof. It has keratin in there. But the lining of the vagina is similar to the mucous membrane lining the inside of our mouth. We don't want it waterproof. We don't want it to be dry and rough like the surface of our skin. We want it to be covered with mucus, because there's still going to be wear and tear there, whether during intercourse, whether during passage of the baby, during the menstrual flow. Menstrual flow has to pass down along here. And so, like any good stratified squamous, it's designed to wear away these layers and protect the tissue underneath. Of course, there's smooth muscle layers. During intercourse, those smooth muscle layers will contract. They will contract outward. It's the same action that they do during childbirth, because the uterus is contracting and the vagina is contracting to help propel the baby, out of the cervix and through the birth canal. Okay. Now this is a breast, and I have a model breast here for you to take a look at. This is used in teaching about breast cancer, and it's been used a lot. But inside here are 2 or 3 BBs that you can feel if you palpate the breast correctly, just like you would do in a self-examination. And that's about, a BB is about as small as a tumor can be detected simply through self-examination. You have to sort of go around like this to try and let me. Okay, I can understand. Sort of like this, you have to palpate around like this to try and detect the presence of the BB. And I know they're in there. I've just forgotten where they are. So I know they're in them, but you can feel them. You can feel them. That's about as big as you can, as small as you can detect something. Anything smaller than that has to be picked up with a mammogram. Now, this is a typical breast. It has a lot of fat cells embedded in there. A lot of fat moves into the breast at the onset of puberty, provide energy for milk production. the milk producing cells are arrayed around the nipple like the spokes of a wheel. The breast is simply a modified sweat gland, extensively modified. The male breast is the same way. The male breast is also a modified sweat gland. The male breast doesn't produce milk, but the structure is pretty much the same. You'll notice that there are a series of little white lines here. These are the ducts leading from the milk glands, all coming out into the nipple. So this is where the baby feeds. It sucks on the nipple and more milk is released. So here we see a lot of adipose tissue, as I said. The nipple is surrounded by a darker structure known as the areola. The areola turns darker during a pregnancy. And then you can see all those little lines there. Those are the ducts coming from the milk glands. And we call them the lactiferous ducts. And we'll see in lecture how these glands are arrayed all like literally like spokes of a wheel. Okay. Now this is the what we call the alveolus. Another term for it's like a follicle where milk production is taking place. This is the alveolus is a follicle or a bubble in here that's filled up with milk. Milk is being generated by these cells located along the edges here. You know, the secretory epithelial cells are secreting milk, and then the milk moves down the lactiferous, lactose or whatever, down these ducts that lead to the nipple. Now, problems with the breast. There are 5 different types of breast cancer that can occur. The most common type is ductal breast cancer, where a cancer grows inside one of the milk ducts. That is probably the most common type. The least common type is inflammatory breast cancer, which looks like a great big lesion or sore spot on the surface of the breast. It's pretty unusual that occurs, but it does occur. Now, the interesting thing here, and we don't know why, is that breast cancer typically occurs more often on the outside upper quadrant of the breast than anywhere else. 50% of all breast cancers occur here, the areas closest to the axillary lymph nodes. Now, we know that the breast cancer, like here we have our breast right here, and this area up here is going to be the primary area for half of all breast cancers. And we always look to see if it's going to spread to the lymph nodes, because the axillary lymph nodes spread down across here under the armpit, and that's where the cancers usually go first when they spread. Don't know why that more of them occur in the upper outside quadrant than anywhere else. Okay, so here we see what breast cancer looks like. The most common type is going to be the ductal carcinoma. The cancer grows in the lactiferous duct, and then it can spread as invasive breast cancer. It can spread to other organs in the body. And you can see down here, this is a duct, and this mass growing here is the cancer cells. Okay. Structurally, take a look at the male structure here in the models. Let's see. Here's a model. There's a male structure. And you've got a female structure there. There. You can pass these around. Here we go. There's 2 female structures around too. Okay, so here you see this is a circumcised penis. The covering of the head of the penis is called the prepuce. That is this region. There's the penis. There's the foreskin. The foreskin extends over the top of the penis, over the end of the penis. There's an opening there. It doesn't completely seal anything off. The foreskin has to be pulled back for urination and pulled back to clean the head. If the patient is circumcised, that foreskin, the prepuce is removed, and you have this ridge there, right where the arrow, the tip of the arrow is. That designates also the area called the glans penis. The glans is the tip. This is the most sensitive portion of the male anatomy. 10,000 times more sensitive to mechanical stimulation and touch than any other area on the male's body. Here we go, body part. The glans penis is analogous to the clitoris in the female. Also in the female's body, spongy tissue shows up in the nipples of the breast. So when the male is sexually aroused, the nipples will become erect, as will the clitoris. Now, so here we see, there's the scrotum, the sac of tissue holding the paired testes. There's one of the testes. There's the vas deferens going up and wrapping around the pubic bone, coming around to the posterior surface of the urinary bladder, and combining with the seminal vesicle. That's the seminal vesicle right there. There's the epididymis, urinary bladder, ureter, okay. Rectum. There's the seminal vesicle sitting directly above the prostate gland. The prostate gland is this little brown structure here. Anus, prostate gland. And so we see the vas deferens combining with the seminal vesicle to form the ejaculatory duct in the prostate. Okay, close up of all that. You can really see the seminal vesicle here and the vas deferens together. There's the prostate. Seminal vesicle, prostate gland, rectum, and anus. And this gives you a pretty good idea of also how easy it is to palpate the prostate through the rectum. So, okay. Scrotum, testes, epididymis. Spermatic cord, that's where you're going to find your cremaster muscle holding the suspending the testes in the scrotum near the abdominal wall or away from the abdominal wall, depending on temperature. Vas deferens. Okay, here we have the cross section. Now you can see this better. There's the glands. There's the prepuce. There's the urethra. We have the corpus spongiosum, and we have the corpus cavernosum. Testes, epididymis, urinary bladder. Here we can see the prostate. And you have the prostatic urethra, and there is the ejaculatory duct. Now it looks like it just goes off and stops. But what it really does is it extends to the outside of the prostate on both sides, because you've got a vas deferens and a seminal vesicle on both sides that are feeding into that duct, delivering semen and sperm for ejaculation. Okay. Here's the urethra. Here we have the testes. Yes, we've seen all this. Okay, female structures. Pubic bone, right there. Urinary bladder behind, directly behind the pubis, the pubic bone. There's an ovary, the fimbriae, the oviduct or fallopian tube or uterine tube. Barely see the uterus in this illustration here. Here's the vagina, rectum, and anus. Okay. Now, close up on all this. Pubic symphysis right there, that's the pubic bone. Urinary bladder, ovary, fimbriae, oviduct, uterus. Let's get into the cross section. Okay. Yes, it's an ovary. Thank you. It's fimbriae. Okay, here we go. Now, you can really see the ovary. Well, you can't see the ovary here, but you can really see the uterus. Notice that the uterus is tilted forward directly over the urinary bladder. There's the urethra. there's the uterus. Now the uterus tilts forward at a 90-degree angle, so it sits right on top of the urinary bladder. Now, in an older female, the ligaments that held that uterus in place get old, get worn. And because typically we say that we have a 90-degree turn here, but sometimes the uterus can tilt backwards. If the ligaments that are holding it in place become worn or stretched, it'll tilt backwards. And in some even more unique cases, sometimes the uterus can slide down through the vagina and protrude out through the labia. It can happen, because there's nothing that, the only thing holding the uterus in place are these ligaments, and those ligaments get stretched, the uterus, and it tilts backwards, it can slide through the vagina. And now you have the uterus projecting through the vaginal orifice. And I had, about 10 years ago, I had two students that were working at one of the local nursing homes, and they had a female patient they were supposed to insert a catheter into. And when they went to do that, you know what a catheter is, right? You stick it into the bladder and you drain the urine out. There was the cervix staring at them, literally protruding through the vaginal orifice. Now, what do? Well, they couldn't do anything, but the uterus has to be put back, because it's a whole lot easier just to push it back up and throw a few stitches in there and hold it in place than it is to open up and have, major surgery to have it removed. So, but it can happen. It can happen. We call it a prolapsed uterus. It's not that unusual, but in a healthy young adult female who's pregnant, you can see why having the baby in the uterus is going to be pushing on that bladder and causing a lot of desire to urinate frequently. So now, other structures you can see, this is the cervix. The cervix is the opening to the uterus. The cervix is designed to protect the uterus from penetration from the outside, because remember, this whole thing is open to the outside. From the vaginal orifice up into the uterus, we go into the fallopian tubes that end in open structures called fimbriae. So anything coming in through the vagina, any kind of pathogen or bacteria that's not pathogenic or debris, could make their way in the abdominal cavity. The cervix tries to take care of that by keeping it plugged with mucus, so that it can prevent the movement of dust, dirt, and debris and pathogens, if you will. Okay, there's the vagina, and this is the mons pubis. This is a layer of fat that develops during puberty right above the pubic symphysis. This is where most pubic hair grows on the female. The axillary hair under the armpits, pubic hair tends to grow on the surface of the mons pubis. Different pattern than the male. Here we see the labia. We have these two folds of skin called the labia major and the labia minor. And there is the clitoris on the anterior end of the labia minor. This inner fold here terminates at the clitoris. Clitoris is this structure right here. When the female is sexually aroused, the clitoris is engorged with blood and stands up as erect. And you'll notice that the vaginal orifice is back here. It is the pressure from intercourse pushing the clitoris up towards the pubic bone that's going to stimulate the clitoris, because the clitoris has to be mechanically stimulated for the female to achieve orgasm. And during intercourse, there's enough indirect pressure to cause that. Now, the outer folds are called the labia major. There's the labia major, and there's the labia minor. And the labia minor, that's going to surround this area called the vestibule. So you're going to have the vaginal orifice and the urethral orifice in there, and it terminates at the anterior end with the clitoris. Okay. And there we have the vestibule. And then the rectum and the anus. Okay. Now just for a little bit of perspective here, we see that this is the cervix, but the folds on the cervix can interfere as well as the vagina with the movement of sperm cells. And again, there's no guarantee that you're going to go in the right direction because you've got two ovaries and you've got two fallopian tubes. And while there is a chemical tracer that the sperm cells can use to track the location of the egg, many sperm cells, since they only have one gear forward, they're going to go up and they're going to hit the top of the uterus, and they're going to bounce off and go down one of these fallopian tubes. And, you know, there's no guarantee there's going to be an egg there to begin with, but if they're going up there and there is an egg, it's likely going to be on one side or the other, and only half the sperm are going to select properly. Okay. Ovary, embryo, secondary oocyte. This area over here is the ampulla. This is usually where fertilization is going to take place. Okay. Uterus, there's the cervix, and there's the vagina. Okay. The cervix surrounds is contained within the vagina. The folds here, there's the cervix right there. This area on either side of the cervix is called the fornix. Many sperm cells that go up in there are going to be stuck. So there's the oviduct, immature eggs, old follicle, ampulla region. Okay, now, okay, here we see the layers of the uterus. We see the endometrial layer. There's the endometrium. The layer closest to the lumen of the end of the uterus is the stratum functionalis. Underneath that is the stratum basalis. Then we have smooth muscle layers in the myometrium. We have the perimetrium right there, the outer layer. Sperm cells, really big sperm cells. Now here we can see the cervical plug. I was telling you about this plug of mucus here is designed to thicken and stay in place through most of the cycle. However, it will thin out somewhere around day 14 in anticipation of sperm that may or may not be present, but it will thin out in anticipation of the delivery of sperm. And then it'll thicken up again after that window has closed to, again, protect the interior of the uterus and the abdominal cavity with this plug of mucus. So, okay. Here we see, of course, a fertilized egg implanted here, right there. Yes. And we call this a blastocyst. You don't need to remember that. You'll hear enough of that in lecture. There's the mucus plug right there. Again, thins out around the 14th day, stays thinned out for about 2 days, then starts thickening up again after that. So, okay. And that is everything I wanted to show you. Now, I do have some good slides. There are some slides with over there that are one is a normal healthy prostate, one is prostate cancer. You can take a close look at. These are real tissues, not just pictures of you can examine. There are some other different. I have a different set of slides at every microscope, so you can take a look at that. And since there's nothing else I can show the people online, I'm going to let you all go. Use your PowerPoints to complete your worksheet. If you have any questions, let me know. Okay.